So good morning, everybody. Welcome back to UAB Pathology Grand Rounds at our new noon time. I hope that works with people's schedules. We had to switch that around a little bit because of some of the scheduling with the residents. So, and you also notice we had planned a schedule where we were actually going to be in person with lunch and a number of visiting speakers, but as of about 2 weeks ago, we scrambled and most of them just postponed till the spring. So hopefully, at least for the external people, we'll have them in person starting in January. But today it's my pleasure to introduce our very own Dr. Jeff Holmes, who's the Dean of the School of Engineering. He started his education out in Hopkins before switching to the other coast, where he completed his Ph.D. at UC San Diego. He then spent a year in Germany studying for his postdoctoral fellowship at the University of Freiburg. He then came back to San Diego, where he completed his medical degree and then moved to the South to finish his clinical fellowship at the University of South Carolina. He started his faculty career at Columbia University, where he rose to associate professor before transitioning to the University of Virginia, where he had a very successful career and continued to work his way up to professor and director of their Center of Engineering in Medicine before we were fortunate enough to recruit him away just a little over a year ago. He was brave enough to come and start a new position as our dean in July of 2020. And he is now both the Dean of Engineering as well as the Goodrich Endowed Chair of Cardiovascular Surgery. As we'll hear in a moment, he has a lot of very relevant work in the area of cardiac repair, where he does both functional as well as modeling work and really leads the way in this area. He's had a number of distinguished awards, including teaching awards, Established Investigator Award from the American Heart Association, as well as a named Distinguished Investigator Award, also from the American Heart Association. In addition to his academic endeavors, he's directed a number of centers, establishing some, raising tens of millions of dollars to complete his work. He's currently on 3 active awards, 2 from the NIH and 1 from the AHA, and has over 100 publications, both primary, editorial, and book chapters. And fitting with his engineering focus, he currently holds 4 patents. So I'm not going to take up any more of his time. So Dr. Holmes, thank you again for accepting our invitation and telling us about scar formation under mechanical load in heart, skin, tendons, and computers.
Thanks so much, Adam. This is the first time I've gotten to do a pathology grand round, so I put some things together a little differently than what I might normally do, and we'll see how it goes. I was hoping throwing the computers in there at the end might get at least a couple of curious folks who wouldn't have otherwise come. So a lot of the time over my research career in my lab, we've been studying infarct scar. And I think most of you probably know nearly 7 million people worldwide have a heart attack every year. And the survivors are at risk for a range of complications, including aneurysm formation and rupture, infarct expansion, ventricular dilation, and heart failure. When I first started in this area, we were interested in the scar because we know that once the muscle dies in that region, it's not regenerated, it's replaced by scar. And there was good evidence already when I was a graduate student that the mechanical properties of that scar tissue are a critical determinant of pump function, remodeling, and these other sequelae that we would like to prevent. And so the simplistic idea was maybe if we understand the mechanics of the scar, we can find a way to modify those mechanics in order to either improve function or prevent remodeling or both. It's a simple sounding idea. It has proven remarkably difficult to do, which is why I'm still working on that same problem 25 years or so after I started. What I want to talk a little bit more about today, or focus on today, is one of the things that we discovered as we were just starting the process of characterizing the scar, looking at its structure, looking at its mechanical properties, is we found that that scar is often highly anisotropic, meaning that the collagens all aligned in one direction. And so these are some pictures These are sliced parallel to the surface of the heart, so parallel to the epicardium, stained with a dye called Picrocereus red and imaged under polarized light. And you can see lots of large, highly aligned collagen fibers, other than lacking crimp, not too different from what a tendon might look like with this same stain. This is in dog and in pig. But then in rat, another common animal model for studying post-MI remodeling, you get randomly aligned collagen. And a few years after we figured this out, we also figured out how to force a rat to make aligned collagen. And I'll tell you a little bit about that as we go along. And so from a mechanics point of view, this high level of alignment was very interesting. And so we focused quite a bit in understanding where that comes from and how to manipulate it. And that's what I want to spend the first sort of 2/3 of the time today on. And the reason to spend time on that, in my view, is that the alignment of that collagen is a potential therapeutic target in the heart, and then as we'll talk about, also in other organs. And it's a therapeutic target for two reasons. One is my previous goal, the original goal of improving mechanical function. We did eventually find a way to do that, and I'm skipping that whole story today, but the references are here. We were able to show that if you could, so this is large anterior apical infarcts, which are the most common one in humans, that if you could longitudinally reinforce that infarct, so just make it stiffer in the longitudinal direction, but not the other directions, that you can significantly improve pump function immediately after an MI. So a big difference, bigger than anything we're able to do through any other method. But interestingly, collagen does not normally align in that direction in the heart. In fact, of all the animal models we've studied, the only pattern we've never seen is longitudinally aligned collagen. We've seen circumferentially aligned, we've seen randomly aligned, we've never seen longitudinally aligned. So this would have to be an intervention to force this to happen, but we have really good evidence that if you could do that, you could make big improvements in function. The other reason to be interested in the alignment is what Jay Zhang does, who's with us on the call, which is regenerative medicine. And so Jay and his colleagues are very good at regenerating dead myocardium. And I don't have to argue with Jay about this, because if my choices are I can have a scar in my heart or I can have rebuilt myocardium, I will take rebuilt myocardium every day, no question about it. However, a lot of the regeneration in practice starts with something that you would do a couple weeks after the heart attack, or it uses stem cells that are going to take some time to mature. And so one of the things that I think is inevitable about some of the regenerative approaches is they're going to kick in at a time when there's already some scar tissue. And when that happens, the alignment of the extracellular matrix is where the myocytes get their cue as they're developing. So they align with the surrounding matrix. And in the heart, it's particularly important that you get all the myocytes aligned the way you want them in order to get proper mechanical and electrical function. And so I think another reason for being interested in alignment of collagen in the heart, even if you aren't that interested in scar by itself, is that it's an important piece of the story of how you're going to regenerate myocardium that has the function that you want. So I'm skipping at least the first decade, which is when I first started my lab, we did everything experimentally. We did animal models. Most of the ways that we studied alignment of collagen were in little collagen gels, so cell-populated collagen gels that we used to study how the cells would remodel the collagen and how they would respond to mechanical stimuli and other stimuli. And to be honest, we weren't getting anywhere as fast as we wanted to with a lot of the in vitro work. And so about the time that I moved to University of Virginia, I was fortunate enough to have systems biology colleagues there in the Department of Biomedical Engineering. And systems biology was an emerging field. I didn't know that much about it. And I learned a lot from them about what's possible in terms of modeling complex systems computationally. And so that's really where our thinking changed about how to approach some of these problems. What we realized at that point was it wasn't that we didn't have enough in vitro data. There was actually tons of in vitro data. We were adding in vitro data to an enormous literature of in vitro data about what governs cell alignment. Where we were struggling was once you get to an in vivo environment where there's multiple cues that are getting integrated at once by the cells, we didn't understand the integration. And so to tackle that, we use something called agent-based modeling. I think some of you are probably familiar with this. There was a really good story about 6 months ago in one of the UAB news publications, about how they used agent-based modeling to design the Spain auditorium setup for the COVID vaccine. So agent-based modeling just means that you model a population of individuals, you know something about how each individual is going to behave, and then you use the model to see what's going to happen to the population as a whole. In our case, our agents are fibroblasts. And in these models, they migrate around, they proliferate, they apoptose. We track the position, orientation, and speed of each cell. And this was really the critical part, that cell orientation is determined by integrating responses to a bunch of the things that we know individually in vitro can affect cell alignment. Things like stretch, which is something that we're very interested in, contact guidance from surrounding matrix, chemokine gradients, and other factors. And then as our cells crawl around, and this is now a color map of collagen density that's predicted by the model, and all the little lines show the orientation of the collagen in each region, we let those cells remodel the existing matrix, deposit new collagen fibers, and then watch how that structure evolves. I apologize. So the reason that we really liked this this type of model, which I had never worked with before until learning about it from my next door lab neighbor at UVA, was it let us incorporate this wealth of in vitro cell culture data that was already out there in the literature. And so this is one example. Many groups had cyclically stretched cells and looked at how strongly a cell aligns with a given amount of stretch. They had published those. We started with fibroblasts. Sorry again. We started with fibroblasts, but eventually added other mesenchymal cell types to this curve. And it's remarkable how consistent they were. People had used the same sort of range of stretches. They had published lots of different alignment measures. We had to go back to the data and sort of recalculate one common alignment measure. The one that we like is a very simple one. It's called mean vector length. You just represent a bunch of angles as a bunch of unit vectors, and then you average them. And so if they're all aligned in exactly the same direction, the average vector has a length of 1. And if they're randomly oriented, the average vector has a length of 0. And so it's a very simple measure of the strength of orientation. And so now we're able to put in dose response curves reflecting a wealth of data for how cells respond to each of these signals that we're interested in. And then the part we had to guess a little bit about was when there's more than one signal and they're telling the cells to do different things, how will the cells respond in terms of their alignment? And so again, here we basically just average the vectors. So if there's a cue from the extracellular matrix saying align this way, there's a mechanical cue saying align a different way, we basically average those and then create a little probability distribution based on how strong that overall orientation cue is. And the cells choose from that. All right, so as our test of the model then, we went back to an experiment that we had done. And this is when I told you that we finally figured out how to make a rat generate aligned collagen in a myocardial scar. And that was, we were using cryo-infarct instead of coronary ligation, so making a freeze injury in different locations as a way of varying where on the heart the injury was. Normally, when you ligate in a rat heart, you basically take out the whole apex. Here, we're able to just do the apex, or we can do something at the equator. And we also wanted to vary the shapes of the areas of damage. And what we found out in this original study was that no matter what the shape of the infarct was, everything at the equator produces aligned collagen and everything at the apex produces random collagen. And so the apparent difference between animal models that we had seen in the past seems to be just a difference in which vessel you tie in order to create those various animal models and where the damage ends up being on the heart. And so in our model, we calibrated one more thing, which is we just adjusted the rate of collagen production and degradation in the model to fit data on collagen accumulation that we had acquired in a different set of rats. And then we turned the model on, put all of the cues in at the levels that we believe they existed in these two different regions of the heart. And right out of the box, the first run of the model matches the data perfectly. It predicts aligned collagen at the equator. It predicts random collagen at the apex. And my usual joke at this point is this is probably the day I should have retired. Because for those of you who do computational modeling, this never happens, right? Especially not a model that was built entirely on in vitro data and then turned loose on a tough in vivo problem, right? And it turns out now, in the model, we can turn off each different factor that we put in there. And it turns out that the mechanics are completely dominating this response. The difference between these two locations on the heart is that they have different mechanics. And we had measured that in the animals, and that's what's driving the result in the model. So now if that's right, we should be able to do something prospectively. We should be able to perturb the mechanics in a way that hasn't been done before and predict what will happen to the structure before we do the experiment. And so we did that, and the prediction that, or the intervention we tried to predict was, what if we sewed a patch on the surface of the heart that prevents circumferential stretching but allows longitudinal stretching? In our model, we would predict that that would produce, for the first time that we would ever see, what we said we wanted therapeutically, which is longitudinally aligned collagen in a healing scar. And this is just some sonomicrometry data showing that we're able to sew the patch on and that we actually can prevent stretch in one direction while allowing it in the perpendicular direction. And when we did that, we got exactly what we predicted from the model. This is average distributions of collagen. So the fraction of the collagen that's oriented at each angular bin from -90 to 90. As the collagen starts to accumulate over time, looked like there might have been a slight preference for 30 degrees, but by the time you get to 6 weeks, really the collagen is evenly distributed enough in the different directions that there's no significant orientation. By contrast, in the animals where we only allowed longitudinal stretch, you get a bias towards longitudinally oriented collagen right away, and then it just grows as you accumulate collagen. So most of this is close to plus or minus 90. So exactly what we predicted from the model. And you know, it's the nature of scientists, right? The thing that bothered me about this is that it all works so well. It just didn't seem right that we spent all this time putting all these different factors into the model and getting the dose response curves. And then when it's all said and done, the only thing that matters is the stretch, right? There's just something about that that was a little hard to understand. I could have made a model that only had stretch in it and ignored all the other factors, and I would have gotten the same result. And so this goes back to why we originally did the experiment with the different shapes, which is we thought that having different shapes of infarcts would create different patterns of chemokine gradient, and that by driving the migration differently with the chemokine gradients, that we would actually influence the orientation of the collagen. And that's really not what happened. Our distributions of the orientations are indistinguishable in the 3 cases. And when we run them in the model, there are also very tiny differences that you can't distinguish. And what we eventually figured out is that if we drive the shape in the model to something that would be basically impossible to create experimentally, so in an ellipse that's got like a 10-to-1 axis ratio, we can eventually see an effect of the chemokine gradients, right? So what's happening here is there are all these dose response curves, and in a beating heart, you're all the way at the high end of dose for mechanics, but you're still basically at the low end of dose for everything else. There are chemokine gradients, but they're pretty modest compared to other systems like skin. And so actually, we were not the first people to make an agent-based model of collagen accumulation. We originally got the idea from a group that had done it in skin. And so now we're showing a side view from their model. This is skin surface, this is deep. And these are the fibroblasts that have migrated in at some point in time. I think this is 7 days of wound healing. And you see that they're predicting these collagen orientations that are essentially just cell migration tracks, that the cells are migrating in and they're laying down collagen that's aligned with their direction of migration. And so they had actually shown that the chemokine gradients were the primary determinant of the collagen patterns, but their model doesn't have mechanics, right? There's no mechanics in there. And in fact, we came back to a similar model using now our system of skin in a collaborative work with a colleague from Virginia Tech named Rob Gordy that just got published, I think last month. And what Rob was doing was they were testing the effect of a drug that influences scar healing. And it's a peptide that they found doing electrophysiology studies in the heart. So it's another heart to skin story. But they accidentally found that it has these other effects on cell migration and scar formation. And so depending on what size your screen is where you're watching me today, you may or may not be able to see that the control, these are punch biopsies from humans that have been treated during healing with the peptide or control. And the control collagen is pretty strongly aligned in the horizontal direction on this picture, which is parallel to the skin surface. This is a side view. And the treated is less aligned. Maybe easier to see in this picture where they've color coded the orientation. Blue is horizontal, and green and purple over here are at an angle to horizontal. And so they've just got a lot more collagen that's oriented at different angles. And what we did with Rob was they had done cell culture studies where they treated with the peptide and tracked migration tracks. We adjusted the migration behavior of the fibroblasts in our model to match those migration tracks. And actually what we matched was statistical properties of how far and how frequently they turned. And then we turned an agent-based model of this problem loose. And initially we're disappointed when we didn't have any mechanical load on the system. We actually didn't see the effects that they were seeing experimentally. We had somewhat disordered collagen at baseline, and we had similarly disordered collagen in the treatment. But once we thought, well, you know, most skin, most places in your body is under a little bit of tension. If we applied just a little bit of tension to the model, then we got into a regime where the mechanics and the chemokines are both playing a role, and you can tilt the balance with the drug treatment. And so in this case, we get that nicely aligned parallel collagen in the absence of the treatment. And once we perturb the cell migration, then we get this more disordered behavior similar to what they saw. All right, so let's stop and think a minute about where we are. So I've told you that agent-based models from our group and others do a really good job of predicting evolving scar collagen alignment in multiple tissues and in multiple different experimental models. In some cases, we've even been able to make prospective predictions about the effect of a therapy that has never been tried before, and then that turns out to be correct. I really like the ABMs because they let you integrate cell level data. And I showed you a couple of examples, dose response curves, some drug effects on fibroblast migration, and then you can predict what's going to happen in a more complex environment with that integration. In the heart, the mechanics just steamroll everything else. You know, it's a beating heart with, you know, high pressures inside, doing this 6 times a second in a rat and 1 time a second in you. And so maybe it's not surprising in retrospect that just sort of steamrolls everything else, but not in the skin. And then the thing that I want to talk about next a little bit more is, so far everything I showed you wasn't mechanistic at the cell level, meaning we took cell culture data, we put it into a model, and we focused on the integration. What I couldn't tell you, for example, is if I treated with a drug, the only way I could tell you what was going to happen is if I went back and collected a bunch more cell culture data with that drug, right? Then I can put it into my model and I can integrate, but I couldn't tell you de novo what was going to happen with the drug because I don't have any of the underlying signaling represented in any of my models. We can do that. And so I'll show you a little bit about how we've started to do that in a couple of examples. This first one, I may be pushing the definition of what you'll accept as mechanism. Normally, when we talk about mechanism, we talk about signaling pathways. This one is a little bit more out there. Here, the purported mechanism is thermodynamics, which is something that only an engineer would try and tell you. So we'll see what you think about this one. But there's this paradox in the literature about cell stretching and alignment that's been around for decades, and it has bothered many people, including me. And I kind of swept it under the rug in the first part of the talk, which is, I told you that we took cell culture data from people stretching cells and then looking at alignment. And then we looked at how strong the alignment was. The part I kind of swept under the rug was all of those experiments were in 2 dimensions. So they'd stretch cells on a stretchy membrane. And when you do that, the cells always align perpendicular to the stretch. But we know that when you do the same thing in a collagen gel or in a soft tissue, the cells always align parallel to the stretch. And so we took the dose response curves, but we flipped the cells 90 degrees because we knew that was what was supposed to happen. Why it is that you get these different responses in these different model systems has, like I said, has bugged people for a long time. And so this was a sabbatical project. I got a chance to take a run at this with a guy named Vikram Deshpande from Cambridge and a guy named Bob McMeeking from Santa Barbara. And what they had built was a thermodynamic model of something that's been known for a long time, which is if you take a stress fiber that's a bunch of actin subunits, if you stretch that stress fiber and hold it in a solution of actin, more actin subunits will assemble into the stress fiber. And if you do the converse and you push it, actin subunits will come out and go into solution. And you're just changing the free energy of whether it's energetically more favorable to assemble or whether it's energetically more favorable to be in solution. And so they took that concept and made a model of cell orientation that basically asked, how do your stress fibers align in the presence of different types of stretch? And so we used that and we added a couple of ideas to it. One is we said, well, one thing that we know that they hadn't worried about yet because they had only been simulating these 2D experiments, but that matters in 3D, is if you put cells in collagen gels or in a soft tissue, especially a loose soft tissue, they can let go, right? They can crawl around, they can let go, they can reattach. So basically the cells can remodel their attachments and they can remodel the surrounding matrix. And no one had really accounted for that, but we need to for tissues. And so we assumed that the cells will remodel the ECM and or their attachments to reach a minimum energy state. And this is where I told you, we have no proof of this at all, and it's a super difficult thing to test. So it's just a hypothesis. And then that allows us to say that in those collagen gels, the boundary conditions that you put on the gel, so if you hang on to the gel in both directions, the cells generate tension, and then they have uniform alignment, randomly distributed orientations. If you only hold on in one direction, fibroblasts will compact the gels in other directions, and they'll align in the direction that you're holding. And so this assumption lets us reproduce all that behavior. And then the piece of the model that they had already worked on is once you start cyclically stretching a cell, what basically happens is if you let a cell shorten very quickly, that's when it starts taking apart its stress fibers. And so if you have a static cell or you're holding something statically, you get this nice distribution of stress fibers where they're oriented. And once you start to do stretching and shortening, especially at rapid speeds, then you lose this orientation and the stress fibers start to orient in different directions. So that's the basic idea. I will not try and put you through all the panels on this slide. This was published in PNAS in 2018 if you're interested. The short version is that we took collagen gels, we placed them under different conditions in the incubator for a couple of days. Then we took them out and stretched them and showed that we could reproduce the alignment behavior from all these different combinations of initial boundary conditions and then stretch. And none of the other models that people had tried to make for cell alignment could do that. And then we found out something else, and this was one where we found it out in the model and then went back and showed that it was actually true by doing more experiments. And this is kind of where my lab is or was. I'm transitioning to more computation and won't have a wet lab here. But this is kind of where we have gotten to as a lab that we used to do experiments and then make a model to understand what happened. Now we do it the other way around. Now we start with a model and we think of that as a really concise representation of what we think we know. So the world's most concise review paper, just a few equations representing how you think the system behaves. And then we do experiments in silico with the model to try and figure out what's the most interesting experiment to actually spend the money on in the lab. And so this was a good case of that, where what we found was that the degree of parallel versus perpendicular alignment that you get depends on the frequency of stretch that you apply, which we expected, but that entire curve shifts up and down depending on the surrounding boundary conditions. So this is what people had reported on stretchy 2D substrates. This is what we found in collagen gels that had different edge conditions, basically. And the reason that there had been this big controversy in the field was that it so happens that the vast majority of people who do cell stretching experiments do them at 1 stretch per second, so 1 hertz. And if you happen to do your cell stretching at that frequency, what you find is that in the 3D gels that people most frequently use, they get parallel alignment. And in the 2D substrates that people use, they get perpendicular alignment. But that's just an artifact of happening to be of where you happen to be on a dose response curve. And so this I liked the model in this case because it let us understand the behavior of the system over a much wider range, and resolve why people were getting these different results from experiments that seemed like they should give you the same result. One more mention in that vein, and again, this is one that's complicated enough. I'm not going to try and put you through all of it, but we went on and took the next step for tendon. So I actually did a split sabbatical. I spent half the time in Denmark at a sports medicine group and half the time in Santa Barbara modeling stress fibers. And the reason I went to the sports medicine group is they had spent the most time of anybody that I could find thinking about mechanical loading, matrix turnover, and matrix alignment in an experimental system where those are much easier to vary than in the heart. It's hard to do mechanical interventions in the heart. There are a lot of things you can do to a heart that make it stop, and then your experiment's done. It's much easier to do mechanical interventions in tendon, and so they had a lot more data than I did. And the paradox in that literature was that about half the people who increased mechanical load on healing tendons in animal models would find that increases collagen content and alignment. And the other half of the people would find that when you increase loading on healing tendon, that you increase collagen content, but you don't increase alignment. And in a tendon, you want maximum alignment. That's the goal when you're healing. And so there's this controversy about whether loading does or does not increase the level of alignment. And so we took a little run at this. My student Kellen made a multi-scale model where he started with a nice model of a rat walking that people at Stanford had developed. That lets him anticipate what the stretches are on an Achilles tendon. Then he feeds those stretches into our cell model to determine what the cell alignment is going to be. We also use just a very simple representation of how that stretch will affect collagen production. And then we integrate that in an agent-based model, just like we've talked about before. And we predict these responses to these surgeries. So this is one way that people do the surgery. They transect an Achilles. They do not repair it with suture. They let it heal naturally. And then they increase the loading by having a rat walk around or even do exercise, as opposed to having it in a cast or using Botox to paralyze the muscles so that you don't get loading. And in general, what they see in these studies with unrepaired is that over time, the unloaded tendons and the loaded tendons both increase their mechanical strength, which is a measure of alignment, but the loaded ones make a lot more collagen. And the model reproduced that. Where the interesting thing came is now when we simulated what happens if you repair with a suture, which is what a lot of other people were doing, we find that in the literature, a suture repaired tendon is the case where loading tends to make your alignment and your strength worse. And that was reproduced in the model. Again, right out of the box without having to match any of the data that we were trying to explain. And the reason for that is the same as what we talked about with the cells. It's because these things are operating on multiple dose response curves at once. And it's just too hard to do the integration in your head and decide what you expect to happen when you're operating on multiple nonlinear curves at the same time. So in this case, what's going on is the unloaded tendons are down here. They're flat, they're not stretching with each walking cycle. And when you suture repair them, they're overall under a little more tension than when you don't. But then once you let the rat start walking, the model rat, you get some increased strains in the ones that are unrepaired, but you get a lot more strain in the ones that have been suture repaired. Any loading you do just induces a lot more stretch because you've tied the ends back together. And it turns out that on the dose response curves that determine cell alignment, only that last case, the suture repaired ones, produces enough strain that it starts causing stress fiber disassembly and poor alignment. Everything else you get about the same alignment. This is the only one that matters. And then both of the loading cases increase your collagen synthesis. So another case where a relatively simple model, Kellen would kill me for saying that because this was his entire PhD thesis, but conceptually speaking, a relatively simple model that's got some dose response behavior in it was able to explain quite a bit of apparently conflicting data. All right. So I'll stop talking about alignment now and just spend a couple minutes here at the end on collagen content, which is the other aspect of engineering scar that we haven't talked about at all. So I've told you that computational experimental studies support a dominant role for mechanics in determining scar collagen alignment in heart and tendon. In situations where it's safe to do it, like a tendon, I think that computationally designed interventions could be used to direct collagen alignment. So you could take a model like the one I showed you and design an escalating loading protocol that gives you the best alignment that you think you can get, given where you are at each stage in healing. And then if you want to change alignment in a scar, the easy thing to do is, of course, the thing that I don't want to do in the heart, which is there are a bunch of ways to knock out fibroblast responses to stretch. And so in these mechanically loaded tissues, if you wanted to turn what is normally aligned collagen into randomly oriented collagen, that one's easy. There's a bunch of stuff you can knock out that will do that. The problem is that that's never what we want to do in these tissues. In both the heart and the tendon, what I want is to direct the alignment to be either a greater extent or even a different direction than what occurs naturally. And that is very tough because the mechanics are driving the normal healing and the mechanics have to be there for the organ to function. And so right now, where we are in terms of the therapeutics of controlling alignment is that we would need to come up with some other way to direct alignment that can overcome what the mechanics would normally create. And that's a tough problem. And that's, big picture, that's probably why we're still working on this problem as long after I first started on it, as we are. Okay, just a little bit more. So we already talked about this, and I'll skip that one to say that that in this review where we talked about this idea of you're going to want to be able to control and manipulate scar as part of laying the groundwork for myocyte regeneration, we both talked about being able to control alignment, which is all I've told you about so far today. But then, of course, the other piece of this is being able to change density, right? And you'd want to do that for a couple of reasons. If you embed, especially immature myocytes in a bed of scar, because the scar is so stiff, it's tough for the myocytes to accomplish much in terms of contraction. And so you'd want to be able to embed the myocytes, get them matured, and then get rid of a bunch of the collagen so that they can really contract effectively. And at the same time, you'd have to pay attention to the fact that their development, their phenotype is going to depend on the extracellular matrix stiffness. We know that that can affect the maturation of stem cells. And so at some point, we'll want to be able to modulate both alignment and density of collagen. And people have tried this before. And so this is another interesting example, I think, from the literature. This is a bunch of data from a bunch of studies in the late 90s and early 2000s, where people thought maybe we should try and increase the density of collagen in a healing infarct so that we can prevent rupture after an infarction. So we could get the collagen to accumulate faster. And they tried a bunch of ways of doing this. Some of these are mouse studies where they knocked out various metalloproteases, thinking that if you could tilt the balance between production and degradation, that you could get more collagen. And then another set of these were different MMP inhibitors that different companies were developing and that they tested. And the remarkable thing was not one of these studies had a significant impact, right? They could never get a measurable change in collagen content, which is an interesting finding. And so just for grins, we one day simulated MMP inhibition in our agent-based model of scar healing. And we just did that by reducing the rate of collagen degradation. Our model is just a single equation. There's a rate of production, there's a rate of degradation. We decrease the rate of degradation and ask what would happen. And interestingly, what happened in our model was you had to wait 6 weeks before even a 75% knockout of all collagen degradation, which I'm sure these drugs weren't accomplishing, would produce something that was outside the error bars of your ability to measure it. And so our prediction all along would have been that that there's no point in looking at any of this at early time points, and almost all of the studies had done it only at early time points. And then our other prediction would be that if you really want to affect collagen content in the heart, you should work on the production side, not on the degradation side. And the reason for that is this. Our one equation that's in this particular model controlling collagen content is the equation that you probably vaguely remember from biochemistry someplace, which is that the rate of activity of an enzyme is the concentration of the enzyme times the concentration of the substrate, right? That's the rate at which you produce the product. And so what happens after an infarct is you get this huge spike in MMPs, that's this gray curve here, early. But at that point in time, there's very little collagen in the heart. Normal myocardium has about 3% collagen by volume. And so you don't get a lot of degradation. Later, after collagen accumulates in the scar, you've got plenty of substrate, but by then the enzyme levels have come way down again. And so again, the product is relatively low. And so overall, this product of the two, which is roughly the rate at which we think the enzymes are degrading the collagen, stays fairly low over time. And so manipulating that is going to be a tough way to have a big impact on the overall collagen content. And so again, this was one of those things we sort of threw out there as an idea. There are many other reasons that those knockouts and MMP inhibition experiments might not have worked. But interestingly, when we sewed those patches on the heart that I told you about, we also did microdialysis in the scar to harvest some interstitial fluid and look at levels of peptides that are associated with collagen production and collagen degradation. And we found exactly what we would have expected from our models, which is that early on, when we sewed the patch on, what we really affected was the production of collagen, and that the degradation effects didn't show up until much later. And so what happened was we changed the amount of collagen that's there to degrade. And so later on, you could see that effect on degradation, but that intervening in degradation in the first week or two wouldn't have made any difference to our results. One other thing we found that was kind of yet another thing that nobody had thought about, I think, until now, is when we measure histologically the amount of collagen, what we're measuring is a density. It's an area fraction. It's what fraction of the pixels in the picture are collagen versus what fraction is non-collagen tissue of some other kind. So it's essentially a density. And to be fair, most ways of measuring collagen, even biochemically, are also really a density. And shockingly to us, even though we knew that the collagen production and degradation rates were a lot different in our mechanically unloaded samples, there were no differences in the density of the collagen. Instead, where the differences showed up was in the wall thickness. In the animals where we mechanically reduced collagen production, we got much thinner scars. And so, going back to those MMP experiments, nobody ever measured wall thickness in any of those. They were all measures of density. And so it seems at least possible that the packing of the collagen in the scar, it occurs in a way that the density stays pretty constant, but it's the total thickness of the tissue that changes. And so it's possible that they were actually having effects in those old studies that were completely missed because all that was measured was density. All right, so just a couple of last slides, and these slides are really fall into the category of things that are possible and things that we would be interested potentially in doing in the future. And this comes back to this idea of mechanism. I told you that when we wanted to embed mechanism of cell alignment inside a model where the cells are crawling around and aligning, one of the problems we had is that those mechanisms actually aren't that well understood. And so we hypothesized a thermodynamic possibility and showed that we can get pretty far with that. But that's never been shown to actually be the mechanism for the cells aligning with stretch. By contrast, when you think about regulation of the amount of extracellular matrix, tons is known about the mechanisms, right? These signaling pathways that are stimulated by many of the molecules that some of you probably study that ultimately result in collagen and MMP production. These are, these are all very well characterized. And so one of my colleagues at UVA, Jeff Saucerman, builds these big network models of all these signaling pathways and how they intersect. And they build the first model based on literature, and then they validate it against the predictions against experiments of different agonists and knockouts that have been done. And this is just to say that that we have started doing things where we can use those models as computational screens. So this is a paper that just came out where one of Jeff's students did computational screens of all the FDA, a library of FDA approved compounds, something like 5,000 of them, and figured out which ones had significant effects just on either collagen production or metalloprotease production. So are any of these drugs that hadn't already been looked at as drugs that you could use to manipulate scarring? What I think is even more powerful about Jeff's approach is there's 5,000 compounds. You could do a high throughput screen for those, but there's also the possibility of combining them in various combinations. By the time you want to do all the combinatorics of 5,000 compounds experimentally, that's going to get really expensive. But you can do it computationally very easily. and then choose which experiments you think you actually want to look at. So I think this kind of approach is very powerful for combinatorics, basically. We also took that same cell model and embedded it in an ABM. So this is just a toy. This is not representing any actual tissue. But what we did was make a grid where we simulate higher and higher levels of inflammatory signals like the interleukins on this axis, and higher levels of fibrotic stimuli like TGF-beta on this axis. And then we let individual cells crawl around. This is a cell's particular migration path. And over time, we can integrate the history of what that cell has seen in terms of its chemical environment and predict how its production of collagen is changing, for example. So at this point, you would need spatial information in a tissue at this kind of resolution in order to do anything interesting with this model. And we mostly don't have that spatial information. And so right now, this is just a concept where you could look at how microenvironments within a tissue are affecting cell behavior. And then one last thing that I'll tell you is, because we don't have that kind of information at the spatial resolution we wanted, we took a run at this, and this is the one that got away. This is the one that got away during COVID. If we hadn't had COVID, or I hadn't moved here in the middle of COVID, I think we were on the threshold. But if anybody does proteomics and you're interested in this and want to take another run at it, I would love to not leave this one where I had to leave it at UVA, which was there's kind of a fun technique called stomp, where you basically go in and anything you can target with an antibody that's got a fluorophore on it, you go in and you map where all those fluorophores are in a tissue section, and then you photo crosslink it's a little less than a cubic micron around each fluorophore, and pull that stuff out for proteomics and get rid of everything else. So you've seen other ways of like scanning slides for proteomics and just looking spatially at what's happening. This one's a little interesting because it pulls out areas that are near a target that you've picked. And our plan for this was to target markers of fibroblasts, markers of macrophages, and be able to detect what's going on in those microenvironments. So, and then we could integrate that back with an agent-based model so that you know what's happening in microenvironments of different cell types, and then can run the whole thing forward to see the effects on healing. And this is just to show that we did some proof of concept with this, and if you plot the level of proteins near fibroblasts versus near macrophages in the early soup, you don't see a lot of differences between macrophage neighborhoods and fibroblast neighborhoods. But once you get out to week 6 of healing, you start to see separations where there are different microenvironments that you can identify around different cell types. And so I think this is a fun idea for the future, but it's one that I had to be willing to let go to move and change jobs. So I'm happy to talk to people about this one if it's something of interest. All right, I'll quit. I see we have a bunch of questions in the chat, and I want to make sure we leave time for a little discussion. So this is an acknowledgement slide with previous postdocs and grad students in the lab, a small handful of undergrads that are more recent. Everybody in red is somebody whose data you saw at some point during this talk, and then also a list of collaborators. I will stop there and see if I can give back my screen so that I can see people while we chat.
Thank you so much for that. As you said, yeah, there's already questions in the chat. Pons, do you want to unmute and ask your question?
Yeah, sure. This is Pons from pathology. I was wondering, in your stretching experiments, have you considered, performing them under hypoxia to mimic the physiological situations?
It's a good idea. We haven't done that at all. We have not looked at hypoxia. So arguably all the conditions I was showing you with the fibroblasts and gels, they have a much richer oxygen supply than what's happening in a healing infarct scar. And we haven't tried to dig into that at all.
Thanks.
Maitri? This one here.
I really enjoyed your talk. I'm not a cardiologist or a cardiovascular biologist, but this was fascinating. I have a question about your stretch studies and the modeling that was done with cells and different matrices, and you talked about the time variations. I was wondering if in your models, is changing stiffness of the cells themselves as a result of change in the substrate a factor? And what are your thoughts?
Very interesting question. That's not in there now. What's in there now is how the stiffness of the matrix affects what's transmitted to the cell. So at a very basic level, when we were doing those thermodynamic models, the big difference between a really soft thing like a collagen gel and a stiff thing like a silicone substrate was that in a collagen gel, most of the stretch that you apply to the edges of the gel never actually gets transmitted to the cell, right? Most of it gets absorbed by the soft collagen, and then the cells only feel a little bit of it. So that's why all those dose response curves were sort of in different places but had the same shape, is that the effective stretch on the cell depends a lot on which matrix it's embedded in. But then you're talking about the next level, which is once the cell starts to assemble its stress fibers, then it changes its own mechanical properties, which will change how it interacts with the matrix. And we did not get that far. It's a great question.
Thank you.
Joanne?
Hey, Jeff, good to see you again. So I really love the modeling studies. Two things, have you added any collagen cross-linking enzymes to these systems to see their effects on the alignment and or the stiffening? And the second question, I'm channeling my inner Tom Barker with talking about provisional matrix, because sometimes you don't see these effects until, the fibronectin-rich provisional matrix and fibrin would have been cleared. So do you think that other components of the matrix can interfere with some of these or complicate these processes? Because you're working with purely collagen in this modeling.
Right.
So there's no earlier matrix.
That's right. Actually, in the agent-based model, I didn't explain this part of it. In the agent-based model, we do start with a provisional matrix, but we don't account for any of the signaling at all. So we do start with a provisional matrix that gets replaced with collagen over time. I was only showing you the density and orientation of the collagen. So it is in there in concept, but it's not doing anything, right? From a signaling perspective, where I think things start to get interesting is one of the last slides that I showed you, where we have cells crawling around through environments that are different. And then we have the underlying signaling network to tell you what we expect that cell to do. That's where I think things will start to get interesting with the different types of matrix, because there you could have fibronectin, you could have laminin, you could have collagens, and you could know about the signal transduction that's associated with the cell binding to those. And so you could start to take into account that aspect of the environment. So far, the toy example we did, we just had 2 chemokines. We didn't try and take into account the rest of it. And in those agent-based models, this is the way we did it for the provisional matrix and the collagen. You can have multiple layers that represent the different extracellular matrix components. So if you know what you think the cells are producing and what they're responding to, you can stack as many components as you want. It's very easy to do computationally. It's much harder to analyze biologically, of course, right? And then with the cross-linking, we have done some stuff with cross-linking in those collagen gel stretching systems, but we did it primarily back on the idea of figuring out how we could modify mechanics in the heart. And this all came from being on a med school rotation where I watched the surgeon, I'm probably not supposed to say this, but I don't even remember who it was, so I can't get him in trouble, where I watched the surgeon dip a Q-tip in a little bit of glutaraldehyde and rub it around the edge of a vascular graft that he was trying to sew to get it a little bit stiffer so that he could sew it in the way that he wanted to. And so we actually worked with glutaraldehyde nitrite, which is a very fast and non-toxic cross-linker. We did a bunch of different cross-linking things in the collagen gels and showed that we could change the anisotropy of those gels. The problem is every cross-linker does the thing that I told you I don't want to do. Cross-linkers take an aligned collagen structure and make it less anisotropic, right? And so I eventually gave up on the cross-linkers because I realized that we figured out at some point our goal was to make more anisotropic, not less anisotropic. And so then we quit with the cross-linkers. One more thing about that, which is it's something I think is fun about the spatial proteomics stuff that I showed at the very end, is, you know, if you pull out cell neighborhoods and then you look at post-translational modifications, you would have the ability to think about cross-linking by different cell types or in different microenvironments. It's one of like 20 things that we saw opening up for the next 15 years for that project. And then I decided to be a dean.
Thank you.
Sure.
Hey, Rikesh.
Hey, Jeff, that was fascinating. And I have a question that follows on some of the themes of the questions you heard already. I love the idea that the modeling informed you on that while the MMP expression goes up, if the substrate doesn't change, what are you really affecting, right? And so related to that, do you know about the Km for the substrate for the enzyme? And then was the modeling based on MMP expression or MMP activity? Because you can change the activity based on cross-linking and oxidative modification of MMP.
Yeah, absolutely. Yeah, at that point, that model was only looking at production, not activity. But that exact question spawned the career of one of my former postdocs. So we knew that a lot of the action, a lot of the interesting action in probably all scar tissue, but certainly myocardial scar, is happening outside the cell after the matrix gets secreted, right? It's all the post-translational modifications and the changes in MMP activity, and then what happens in terms of how it's processing the matrix. And so, Will Richardson, a former postdoc of mine that's now at Clemson in bioengineering, that was how he started his lab, was building network signaling models like what I showed you for the inside of the fibroblast, but building them of all those MMP cascades so that he could start to ask those questions about how interventions are affecting what's happening in the matrix outside the cell. So that's, it's obviously a very complicated system, but trying to map the whole system or big parts of the system has the same advantage that there are so many interactions between all those different metalloproteases and all the different things that they cleave each other, they activate each other, they deactivate each other, that unless you're willing to build some sort of network instead of just focusing on one protein at a time, it's really difficult to get an integrated handle on what's going on. And so I think those are the kinds of problems where the systems biology, the systems modeling is just absolutely critical. It's just too complicated to do it one protein at a time.
Yeah.
Okay, Grish, I think you raised your hand.
Indeed, very wonderful talk. I really enjoy it. The question I have on the slide you have on fibrosis signaling, and you said you have validated those genes. I want to know how you did the validation. And that means if you knock out those genes that will lead to fibrosis, or that how that affect cardiovascular physiology. That's what I want to know.
Yep. So the way those were done is Jeff's lab reconstructs the signaling network from papers that basically just give him connectivity, right? So you gather hundreds of papers that say this thing phosphorylates this thing phosphorylates this thing dephosphorylates this thing, and you line up all the connectivity. But back to Rakesh's question, without any kinetics at all. It's just the connectivity, right? Then you turn this thing loose, and the predictions that they make are, if I give an agonist or I give an antagonist or a blocker of one of the pathways, do I correctly predict whether the gene expression of the collagen or whatever other species you're interested in goes up or down? So it's not a quantitative validation, it's an up-down qualitative validation. And you know, Jeff had a hard time selling this to reviewers at the beginning, because the natural instinct when you think about signaling is, well, if you don't know the kinetics of those reactions, how can you ever do anything, right? And the reason that model works, that you can get a lot out of the structure, not everything, but a lot, is because, at least this is our current understanding, is that most of the things are operating near their EC50 in vivo, right? In a live cell. And because most things are operating near their EC50, you can make pretty good predictions about whether an intervention is going to push you up or down, right? I can't tell you what concentration is going to give me the maximum response, because that is a kinetics question, but I can tell you up or down. And so that's the fundamental insight, I think, from his models that has allowed him to do so much more with just network connectivity than what people had expected would be possible.
A great discussion. Silvio asked to ask a question, and then we have two quick ones in the chat, but we're approaching the hour.
A fascinating talk. Healing of the infarct takes a long time, several weeks. At any given moment, there is a lot of heterogeneity in the area of infarct, periphery against the core, et cetera, et cetera. So any strategy that you are thinking to align the collagen fibers, when and where would you start doing that?
Yeah, that's a fantastic question. And the heterogeneity is something that I didn't talk about at all today, but it's been a whole nother arm of the lab. The best intervention would be something like our patch, right? That patch that we sewed on that altered the mechanics, that started reorienting the first collagen that was laid down, and then it kept reorienting everything else, right? So you didn't have to worry about the exact timing of it. It was changing all the collagen, right? The early collagen was aligned, the later collagen was aligned. And so if you're trying to do this with a chemical or some intervention that has a short time window, I agree it's going to be really tough, right? Because you'll affect you might affect the border, but not the center, or you might get the center, but not the border. And so it is going to have to, whatever intervention we use is going to have to be durable over some period of time. And that's why the patch was great, because that was a durable intervention. It's just one that I don't think is translatable.
Will it be from epicardium to endocardium, will align everything in the same way, in the same axis?
It does, actually. The mechanics are pretty uniform across the wall. But there's one other thing that I didn't admit to you, which is in our model, the cells crawl in from the wound edges. And if you assume that they're crawling in from the wound edges, right, rat infarcts are quite big compared to the heart. Under that assumption, the fibroblasts don't get to the center anywhere near as fast as they should. And so what people think is actually happening with fibroblasts is a lot of them are coming from progenitor cells in the epicardial layer, and they're crawling down transverse to the plane that I'm modeling. And so there are definitely some limits in terms of the heterogeneity that we're not capturing yet. Whereas if we did kind of a 3D stack, we could have things like the transverse migration. That's not in our models.
And the last two questions, if pH is in your model, and if you could if you could give surgeons any advice based on your modeling.
Yeah, so the pH, we have not measured it, which was a missed opportunity because we sampled all that microdialysis fluid, and we used the microdialysis fluid to measure cytokines and things, but never checked the pH. So sorry on that one. That's a great idea. And in terms of the advice for surgeons, I've tried to be really careful about that. The one that we thought we really had a good handle on was this idea of a surgical reinforcement. The one where we originally showed that longitudinal reinforcement improves function was a study with a surgeon. And we were at we were talking to a couple of companies, and we're at the chronic large animal trial phase when that one finally failed in terms of its translation. And the reason was, part of why we decided to make all these agent-based models is that we were defeated by remodeling. And this is a challenge in the heart, right? Anything, and in a lot of these organs, anything you try and do, it's going to remodel in response. And so you have to be able to predict the remodeling and aim for where you want to go, accounting for the remodeling. So what happened with those patches is we sewed patches on the epicardium, and our surgical colleagues felt that there would be a patient population that was getting open bypass grafting where that would actually be a viable thing to do, right? Not an acute infarct a couple of days after you just had your infarct, but somebody who had an infarct that was getting a bypass graft. And so in the chronic trials, what happened was the scar underneath the patch started shrinking as it was remodeling. And over time, it unloaded the patch that we had sewed on. So you got this huge functional benefit at the beginning, and by 8 weeks, it was gone. And so I don't think that I'm ready to tell a surgeon to do anything yet, because I think what we really understand is how complicated this problem is. But I do think we're armed with much better computer models that have reached the point where we can make predictions that either are correct or at least are interesting enough to help decide what's the next experiment that's really worth spending time on. And so I do think we've gotten to that point. And I really did flip my lab and tell the students, you can't do an experiment until you show me in at least a toy model why it's an interesting experiment and why that's better than the 10 other ones that you could do. And that has really changed our thinking about how to spend our time on the wet side of the lab.
That's great. Well,
Jeff,
thank you.
I'm George Nero. I'm sorry I joined late, but definitely enjoyed the parts I attended, and belated welcome to UAB.
Thank you.
Biomedical engineering department is a very close partner for us, and a lot of endeavors and collaborations. So we're thrilled. Thank you.
Thanks so much, and thanks for having me today. It was a great discussion.
Okay. Well, thank you all, and thanks for the great attendance, and hope to see you all next Thursday at noon. Thank you again for the outstanding talk.
Bye, everyone.
Thank you, Adam.